Guys, and welcome to another lesson for A Level Biology. This is Mr. Phillips. Today we're going to be looking at the different types of immunity, vaccinations, and then monoclonal antibodies. So let's go for it. So the last time I spoke to you, if you haven't watched the immunity or the immune system video, we talked about what antibodies are. So we're not going to be looking at antibody structure or how they work today. But just to remind you, when you talked about the natural active immune system, we said that you have an initial exposure, so you get ill because you have very few antibodies in your body. And then the antibodies produced much more rapidly upon the second exposure and to a higher level. And obviously, when we're doing vaccinations, we're just doing an artificial version of this. Instead of getting ill, we're inserting an artificial form or artificially inserting and naturally, I suppose, injecting antigens into our body somehow to stimulate our own immune response. So just to remind you what an antigen is, it is a protein that is on part of a foreign molecule. So it could be a toxin, it could be a pathogen, and our white blood cells recognize it as a foreign material, so material not present normally in our body. Then what we do is we basically deliberately expose ourselves to this for vaccinations. So vaccinations is artificial active type of immunity, sorry, because we're artificially inserting a pathogen into our body which is active because our body still has to make the antibodies. There's the deliberate exposure to antigenic material that has been rendered harmless, and it activates our immune system. So what happens is we insert the pathogen or the antigen into our body. Our white blood cells carry out the response, so you'll get phagocytosis followed by the T cell lymphocytes and the B lymphocytes, and eventually they'll differentiate to make plasma cells, which will make antibodies to stop the virus. And then if the real form of the pathogen ever comes into your body, you won't ever get the symptoms because you'll rapidly, as that last graph showed us, produce the antibodies and you won't get any illness. So what is in the vaccine? Well, it could be the whole microbe. It could be something that's very similar to the living one, but is less harmful. So, for example, when we first came up with vaccines, Edward Jenner injected people with, or exposed people to cowpox, which was very mild, but it stopped you getting the deadly smallpox. It could be a harmless version of the pathogen, so there might be like harmless versions of it, which we can inject ourselves with. Might be a weakened version, might be a dead version. It might just be the antigens taken off the pathogen, or it might be harmless toxins from the pathogen as well. And obviously they can be injected or orally given, or sometimes now breathed in the nose, as in the flu. There are two types of vaccines, and so they basically you need to know more about herd vaccinations. The herd vaccinations is what they were initially thinking about with COVID-19, but they decided it wasn't going to work. And that is the idea that if enough people are vaccinated across the population, so let's say 80 to 85% was needed with smallpox, but for most diseases it's above 90%, then the people who haven't got vaccinated won't be able to catch the condition because there aren't enough people who can carry the condition in the population. So if I've been vaccinated, let's say, against measles, and you haven't, you cannot catch measles from me. Now, the other one, which is much more used in cattle particularly, is ring vaccinations. Let's say there was an outbreak up here of foot and mouth disease, another one here, but then we've got completely healthy stocks of cattle, let's say, over here, and I know down here, something like that. Okay, the idea is what you would do is you would, with the fields of cattle between, you would vaccinate like all of the cattle in this blue area here, and probably obviously into some of the green areas as well. And that means then you'd have like a ring of vaccinations so the disease that couldn't travel from one place to another. Obviously that's only going to work with things like cattle and livestock where they live in the confined places. Obviously with humans that is very unlikely to. So then there are four types of immunity which you must know, often overlooked because it's simple, but people do struggle with it when it comes to questions. So active immunity, okay, is when the body must make its own antibodies. So it's immunity developed after contacting a pathogen or antigen in the body, and you have to make your own antibodies. So first one is natural infection. Okay, so natural active immunity or active natural immunity, that's when you get your infection, your white blood cells go down the series we discussed in the last video, and they then make you, next time you get exposed, you won't be ill again. Other one is artificial active, which is what we've just talked about, which is vaccinations. And vaccinations obviously are when you would have had vaccinations for like MMR, flu jabs, things like that. But the ones that people sometimes overlook, you don't need to know as much detail about these, are the passive immunity types. So passive immunity is when you are provided with antibodies. So you don't have to make antibodies. They come from outside your body, those antibodies or antitoxins. And there's two different types. So there's natural immunity, okay, which is the best example is from the mother to the newborn baby or the unborn baby. So it could be in the placenta where they pass antibodies across the placenta from the mother to the developing embryo, or in the mother's milk when the baby's born and they don't have a very good immune system for first few days and weeks, then it gives them a temporary supply of antibodies. It's only temporary. Other example would be another type of immunity is artificial or passive artificial immunity, and that is when you would inject again antibodies or antitoxins. And we would do that for something that you don't have the time frame to produce your own antibodies. So, for example, if you got bitten by a snake, anti-venom is going to work. Is antibodies are given to you as quickly as possible, then it works straight away. You don't have to make them yourself because you wouldn't survive long enough. Tetanus is another one where that is often used because tetanus is an antitoxin and basically kills off the bacteria which causes it, so that you don't die in the time it takes your body to fight it off, because obviously it's quite a serious infection. There are issues with vaccinations. So vaccinations, some people don't respond very well to vaccinations. There are lots of side effects seen because some people have allergies and your body thinks it's an allergy. Some people who have malnutrition don't suffer well from it. And if you've got an autoimmune deficiency disease, your white blood cells may not be able to make the effective antibodies against it. People have been vaccinated might actually pass it out, which isn't great. Okay, so that's not what we're working as antigenic variation. Obviously, there's ethical issues. There's these anti-vaxxers and things like this, so people don't believe it's safe to do, and that then causes other problems because people in the environment don't have the vaccine. So lots of potential ethical issues. And obviously, the testing is also an issue. How do you test vaccines? Do you test them on animals or volunteers? Is that safe? The issue with obviously some vaccines is that you have to have a new vaccine each year for like the flu jab, for influenza virus, and that is because over the space of a few months, the flu virus will mutate and its antigens will change. So the antigens for the most common strain of the flu last year may not be the same for the common strain of the flu this year. So when we give vaccines every year, the vaccine is developed for the most recent strain of the influenza virus with its antigens, but it will differ a year later. So let's go to monoclonal antibodies. So monoclonal antibodies obviously link into the antibody stuff we did last time. They are produced, you should know this from GCSE, but the idea of monoclonal antibodies is you produce millions of the same antibody, which we could use for different reasons, different treatments of disease and things like this. The idea is with this one, how do we actually make it? Well, a mouse will be vaccinated with whatever it is that we want to make the antibodies for. So let's say we were trying to make antibodies to kill to attack a cancer cell. We can inject them with the cancer markers, the antibodies from that cancer cell to the mouse. Whether the mouse can deal with it or not, it would start to make antibodies for that. Okay, and we would get those from beta cells in the spleen. We then take those cells and we fuse them together with a tumor cell because they continue to keep developing and keep dividing continuously, and that forms something called a hybridoma cell. Now, the hybridoma cell then it can be grown in the lab, and we can take the antibodies that are produced from it. So let's talk about what they are first of all. Sorry. So antibodies are produced from single form of genetically identical beta cells are called monoclonal antibodies. Mono meaning one, clone meaning lots of the same type. We make antibodies which bind to whatever substance we need, and it could be antigens, but it could be to hormones and things like this. It depends what how we shape them and how we make them. Each clone of the plasma cell only makes one type of antibody, so it's a one mono, and as I said, lots of them made to clonal. How can we use them? Well, we could use them to look for deficiency diseases. So it could be if they either attach or don't attach to the presence of some hormones. It could be to look at cancer delivery of drugs, and it could be used for testing of diseases, as we're going to look at. The cancer treatment here, you can see an example of cancer treatment, potentially how it can be used at the minute. Obviously, chemotherapy has horrible side effects because it damages any cells in the surrounding area that are dividing. Whereas if we developed monoclonal antibodies that had specific receptors that fit onto the cancer cells antigens only, it would ignore and it wouldn't attach to any of these normal body cells, but it would attach to the cancer cells. And if you then put small amounts of the anti-cancer drug onto that monoclonal antibody, it will only deliver that drug to the cells in the cancerous cells. So therefore, if you, let's say, had liver cancer, you would unlikely have your hair fall out as a side effect because the cells up here are nowhere near your liver as a result. So it would reduce those side effects of the neighbouring and the whole body cells. We also think it could be used for magic bullet, they call it, drug delivery. So for example, really again the same as with the cancer cells, is that idea of just getting it into those cells that need it and not affecting any of the other cells. And it could be useful for different conditions like cystic fibrosis and things like this, where you just want it to attach to the membrane cells. We can also use monoclonal antibodies for pregnancy testing, and obviously that's something that is used. And the idea of this is that as soon as you become pregnant and the embryo implants into the uterus, the female starts producing something called human chorionic gonadotrophin hormone, or HCG. And it's the only hormone, because it's a very small hormone, lower than 69,000 relative type mass, that leaves your kidneys and comes out in urine. So how this works basically is this: if you got a question for this, you'd be expected to say, first of all, you'd have to urinate on the stick, you'd release HCG if you are pregnant. So most of the pregnancy tests work either using blue or red beads effectively. So what you would have is monoclonal antibodies that have a bead attached to them. So you probably can't see it that well on this diagram, but there's a bead sort of there, and a bead sort of there, and a bead sort of there, etc. Okay, now if the hormone attaches to the antibody, the hormone and the antibody then will form a HCG antibody hormone complex, and they will start to move up the stick. Okay, and if they move up the stick, what will happen is they hit a row of immobilized antibodies, which basically will then also attach to the human chorionic gonadotrophin hormone. So if you imagine that this green circle here, which I'm drawing onto here, is the hormone, okay? If you do have the hormone, that antibody with the blue bead will move up the stick, but it will then attach to this second antibody here, which keeps the original antibody with the blue dye there. So what you then get is you get a higher concentration of these antibodies with blue dye in one area. So if they were to be building up all along here, eventually what you get is a blue line which tells you you're pregnant. If you're not pregnant and you haven't released HCG, the antibody without the hormone just passes through because it doesn't attach to the immobilized level of antibodies here, because it doesn't have the hormone. So these complementary receptors will not attach to the hormone, and they will go up, and it will show you that ones that haven't got the antibodies have passed, and that will cause you a control line in the pregnancy stick. We then have something called, we use monoclonal antibodies for ELISA testing, which stands for enzyme-linked immunosorbent assay tests. And basically what that is a good way of testing for diseases, or as we said earlier, potentially you might have deficiency in something. And we're going to look at the two sort of methods now looking at this. So there are two types of ELISA tests. The first type is a direct ELISA test on the left-hand side of the screen. And this one is used to test for particularly, again, for deficiencies, but also for cancer. So very useful in the detection of prostate cancer in males. And basically how this works is this: the patient's sample is attached to the bottom of a plate, okay, and that'll obviously have the antigen for the disease if that person has the disease. They then add an antibody, which is complementary, as a complementary shape to the antigen, to the sample, and that antibody will have an enzyme attached to it. Now, if the person has the disease, that will attach and you'll get an antibody antigen complex. If the person doesn't have the disease and you don't have the antigen present, that won't attach. So then what they do is they wash out the well of the ELISA test, and if you do have the disease, that antibody has still attached and therefore it will not move away. If you don't have the disease, it's washed away and therefore it's no longer going to give you a positive result. They then add a substrate to that, and if the person does have the disease and the antibody is still present, which is because it's attached to the antigen which was present from the person's sample they gave, that means then the substrate will react with the enzyme that is attached to the disease and it causes a colour change. So that is that. Now, the different way of doing that is then indirect ELISA testing. So indirect ELISA testing is particularly useful for the HIV antigen testing, and it is pretty much the only example you're going to be asked about this, but it is obviously a really, really important one, because if you're tested for HIV and you know you've got it, then that's going to help stop the spread of the disease. So how does this work? Well, this one is a bit different in that this case, we, rather than like, if you remember, go back to direct, we put the patient's antigens in there. This time we're looking for the antibody from the patient that's naturally found in your blood if you have HIV. So we put an artificial antigen into the bottom of the well. So this has been put in. We then add antibodies that are from the patient, okay? So we add antibodies that are from the patient. Now, if you have HIV, even though we can't kill it off, even though we can't, it's, you know, never going to be cured in terms of naturally from humans, you will produce antibodies for it because your white blood cells are trying to make antibodies all the time. Now, they're specific enough that if you add them in, they will attach to the antigens that have been put on there, and they will stay attached to that after we wash it out. Obviously, there'll be other antibodies in your body that do not have any specificity to HIV. So if we then wash it out, if you do have HIV antibodies in your blood, they will stay attached, whereas the other ones will not, and that is obviously a sign you've got HIV. But how do we then detect whether we've actually got HIV? Well, you can't attach anything to the HIV antibodies, like a colour or anything like that, because obviously they're naturally coming from your body. So we add a second antibody, which has an enzyme attached, and this antibody will attach itself to the HIV antibody if it's still present and it's still attached to the antigen, and that would obviously only happen if you do have HIV and you have the HIV antibody. So you can see here this yellow line, bit vague around it, that's got the antigen, it's got the HIV antibody, which is coming from your blood sample as the patient if you are HIV positive, and now we've attached the second antibody, which is complementary to the HIV molecule, and therefore it will attach to the HIV molecule. Now, onto that second antibody is a enzyme, which you can just about see there, okay? And the enzyme, again, will react then if they add a substrate, so they'll wash that all out again for a second time. So anything going to attach will not stay in there. And then they'll add a substrate to that. The substrate will cause a reaction with the enzyme, and again, that will lead to a colour change if you are HIV positive as a result of that. So again, really useful test there because it allows people with HIV to identify they've got HIV. And obviously, if you're then in a relationship, that makes you think, okay, hopefully you need to have protected sex. If you're a drug user, it will obviously give you advice about needles and sharing needles. And obviously, if it was for a blood test, if you're giving blood, donating blood, these tests will be done on people that don't give blood donors to make sure they do not have HIV. And obviously, as per usual, there are ethics involved with this. Whilst there are many uses of monoclonal antibodies that are really important, so from pregnancy testing all the way to HIV testing and cancer tests, it is obviously an ethical issue, and that is because at the end of the day, we have to use animals. We have to, at the moment, to produce these monoclonal antibodies, and this is not always as straightforward as it might seem. The animals could suffer as a result of being involved, or do suffer because obviously the mouse has been injected with all sorts to make the monoclonal antibodies, and they have no choice of being involved as well. Okay, guys, I hope it's been useful, and I'll see you again soon. Thank you.